snowshoe hares. So here on the x-axis again we have time, and on the y-axis we have percent browsed. The red lines are birch and the blue are willow, and the dashed lines are partial browse, whereas the solid lines are fully browsed. So these were browse transects done by Dr. Krebs and his colleagues. And you can see that in the snowshoe hare peak years that there is also a peak in shrub browse. So here in the 1990 peak, the birch were browsed above 80% along the transects. So this is a significant impact on the shrubs. So I'm testing herbivory in years where there was a low of snowshoe hare density and also a low of browsing, and then years where there was a peak of snowshoe hare density and also a peak of browsing. So this is sort of the natural experiment of my project. With regards to nutrients, part of the Kiwani project experimentation was fertilizing kilometer squared plots with a plane. So kind of crop dusting the boreal forest, if you can imagine that, with a standard NPK fertilizer. So I went back to these grids and sampled shrubs in control and fertilized grids to see if there are lingering effects of this nutrients. So I'll be able to test in years where it was fertilized and after, and up to 20-some years after, to see if nutrients have had a big impact. And lastly, climate. So temperature is generally increasing, growing season is lengthening. So by using weather station data and other climate metadata, I can test and see if climate is having a bigger effect than the other two factors of herbivory and nutrients. So is it bottom-up effects, is it top-down, or is climate overriding those traditional effects? So I did a biomass survey in the spring, of which I'll share some preliminary results. I took raw measurements of hundreds of shrubs, and then lastly, the main component of my research, I sampled stems, by which I'll do dendrochronological analysis on. So here's an example of a willow on one of the fertilized grids in a thin section. So the shrubs are very small, so unlike trees that you can core, you have to take a section of the stem and then you use something called a microtome to thin slice it. So here is the sledge microtome that I used. You could see that the blade does a very thin slice of the stem, and then you place that stem on a slide and take a photo with a microscope, and that's how you end up with a photo that you're able to count on your computer. So preliminary results. Here we have the box plot on the left is during the fertilization years, and then the box plot on the right is after the fertilization years up to 1996, and then plus the year that I went back last summer. So you can see the fertilized grids, which are the green boxes. One of them in particular has greatly increased in biomass, and then the other two grids are the control, silver, sulfur, and they've also increased in biomass, although not to the same degree. So you can this tells me that shrubs are increasing through time in biomass, but that in particular fertilization had an impact on this factor. And here we have just generally the biomass has increased through time, and that there's huge spatial variability, but you can see that biomass of, this is birch in particular, is increasing through time. So this sets up the next stage of my research, which is the analysis of these growth rings of shrubs. So now my next stage will be to analyze the dendrochronological data to tease apart what factor is causing this increase in biomass. So stay tuned for more results. The analysis will be taking place, and I'll hopefully be publishing next fall, winter. So basically, the boreal forest is changing structurally. Climate change is impacting in many different ways. There's altered disturbance regimes. Trees are encroaching upon things like grass meadows, and habitat is changing for things like snowshoe hares and other key wildlife species. So thank you very much for listening. There's more info on my website, megangrabowski.weebly.com, and many thanks to the local First Nations, my committee, my field assistants, and all my funding support.
Thank you very much, Megan. In the interests of time, save your questions, and we're going to move on to the next speaker, who's Raisa Philibert, who's going to talk about the effects of iron and light co-limitation on Southern Ocean phytoplankton. Are you ready to go, Raisa?
Hello?
Yes.
Does it work?
Yes, it does.
Okay, great. And can you see, let me see, is my screen, let me just make sure that my screen is up. And is my screen up?
Yeah, but
can you see the slide?
Yeah, but it's not.
All right, great. So
yeah, that's it now.
Okay, that's it. Okay, cool. So as you've just heard, I'm going to be telling you about the effects of iron and light on Southern Ocean phytoplankton. And this is work that we just did on a cruise 2 months ago. So first, what are phytoplankton? So phytoplankton are unicellular microorganisms which photosynthesize. They live in aquatic environments, and as you can see from the picture, they exist in a variety of species, in a variety of shapes and sizes. And what makes them so important? Well, the first is that phytoplankton are the base of a marine food chain. They take up nutrients, and zooplankton graze on them, and higher predators will graze on the zooplankton. And phytoplankton also